Hi there, my name is Mandy Game, and I'm an undergrad at the University of North Florida where I study body plan evolution in tardigrades with Dr. Frank Smith. My research is focused on tardigrade appendages, the genes that pattern them, and how they got so small. The first thing I looked at in tardigrades were the leg gap genes, and through that study found that tardigrades are missing a very important gene called Dachshund, which patterns the intermediate regions of appendages in other animals. This led to the conclusion that tardigrades have lost an intermediate region of their appendages and have a reduced proximodistal axis. To learn more about leg gap genes in tardigrades, we published a paper about it this past summer, and the information to find that paper is right here. Next, I looked at the distal components of leg patterning. I discovered that while many of these distal patterning genes found in tardigrades' closest relatives are present in tardigrades, the expression patterns of those genes are very different in tardigrade legs. In fact, only this gene aristaless appears in all 4 distal tips of tardigrade appendages during the developmental stages we have investigated. This suggests that the distal patterning network has also been secondarily simplified in tardigrade legs. More recently, I've been focusing on upstream components of tardigrade leg patterning. In other animals, the gene SP69 is an activator of Distal-less, this important leg gap gene. My in situ results show a pattern that is very similar to that of Distal-less at these developmental stages in tardigrades. Other components to consider are the Wnt genes. Here I have confocal images of Wnt5 and Wnt16. These experiments were performed by my lab mate, Raul Chavarria. He is a very cool scientist, and you should go check out his poster about Wnt genes in tardigrades. Wnt5 shows broad expression across each developing tardigrade limb, while Wnt16 is expressed in these ventral leg stripes. All of these patterns are consistent with what is found in other animals with appendages, but we will need to do more experiments to determine whether these genes are truly upstream in the leg patterning network of tardigrades. As you can see, while many of these genes known for patterning the appendages of other animals are present in the genomes of tardigrades, the expression patterns of these genes tend to be very different. The predicted upstream components so far appear to be more conserved in their expression, but things get messy when we start looking at the predicted downstream components. This supports the hypothesis that tardigrade appendages are secondarily simplified beyond just their reduced proximodistal axis. In the future, we hope to use RNAi to determine the functions of these genes and to see if interactions between genes such as SP69 and Distal-less are conserved in tardigrade legs.